This video demonstrates two key techniques for sampling and processing biobank porcine tissue specimens. The submersion technique reliably estimates total organ and tissue volume, and the orientator technique generates random isotropic tissue sections for unbiased estimation of quantitative morphological parameters.
The main advantage of the submersion technique for tissue density determination is that it can be applied to any kind of tissue and that it produces reliable results for small as well as for large samples. The advantages of the orientator technique are that it is highly efficient and also works with large tissue samples.
To begin volumetry for a large organ, excise an appropriately sized piece of tissue from the organ. Carefully swab the sample with a paper towel to remove excess blood and tissue fluid. Then weigh the sample on a precision scale and record the weight of the sample. Add 0.9% saline to a beaker of suitable size. Do not completely fill the beaker to prevent overflow during tissue immersion. Place the beaker on the scale. Then, submerge a forceps or a self-constructed sample holder in the saline until the mark on the forceps or sample holder reaches the liquid. Next, reset the display of the scale to 0. Carefully attach the tissue sample to the forceps or sample holder and completely submerge the sample in the saline until the marked position is reached.
To record reliable density measurements, it is important to ensure that the sample is completely submerged and to avoid any contact of the submerged sample to the inner walls or the bottom of the beaker.
While holding the